Hello everyone, welcome back to the class. In our previous class, we had learnt about translocating or targeting of mitochondrial proteins to the mitochondria, especially to inner membrane and outer membrane. And also we had revised the topic of targeting of mitochondrial proteins into the matrix. So let us now revise one by one. So first we had learnt about targeting of protein to the matrix of mitochondria. So it all begins with the protein, matrix targeting protein binding to Tom complex. The Tom 20, the protein which is having N-terminal cleavable matrix targeting sequence is recognized by Tom 20 of the Tom complex. It recognizes this pre-sequence and transfers the precursor protein to central Tom 22 complex. After that, it is directed to TIM23 complex, which remains associated with PAM. PAM is having MTHSP70 co-chaperones, nucleotide exchange factors and other proteins. This MTHSP70 binds to this protein or the polypeptide chain and drives its movements to the or into the matrix with the help of ATP hydrolysis. After the protein has entered to the matrix, that protein gets passed on to another chaperone that is known as HSP60, which converts the unfolded protein to proper folded protein. Next, the targeting of mitochondrial proteins to the inner membrane. There are two types of proteins which are to be translocated to the inner membrane. They are single pass and multi-pass. First, we will take single pass transmembrane protein. The single pass transmembrane protein are having N-terminal cleavable matrix targeting sequence, which is also having stop transfer sequence. N-terminal cleavable matrix targeting sequence is recognized by Tom 20 receptor and it moves to TIM23 complex. After recognizing and after moving through TIM23 complex, the stop transfer signal arrests the translocation so that the protein remain in the inner membrane and being released laterally into the lipid layer. After the protein moving to the TIM23 complex of the inner membrane, it moves to OXA complex where it mediates the insertion of those inner membrane protein in the inner membrane of the mitochondria itself. Next is multi-pass transmembrane protein. The protein having internal targeting sequence which is recognized by TIM22 complex. The TIM22 complex, before getting into the TIM22 complex, the ITS or the internal target sequence is recognized by TOM70 receptor. TOM70 receptor. After that, it moves to TIM22 complex. Then, targeting of mitochondrial protein to outer membrane. The proteins which are to be targeted to the outer membrane are beta barrel protein. First, it is imported through TOM complex. Imported through TOM complex. Then it interacts with TIM complex and are inserted into the outer membrane by SAM complex. SAM complex, which is present on the outer membrane. So today we will be learning about plastids. It is a double membrane bound semi-autonomous organelle present in all living plant cells and photosynthetic protists. All plastids contain multiple copies of double-stranded DNA as genetic material and 70 as ribosomes for protein synthesis. Plastids are differentiated from proplastids. The collective term used for different kinds of organelles derived from the proplastid is known to be plastid. Proplastids are inherited with cytoplasm of plant egg cell. Immature plant cell differentiates and proplastids develop according to the need of the specialized cells. They become chloroplast, leucoplast, chromoplast. See here, proplastid is a progenitor cell and proplastid can become chromoplast, chloroplast or leucoplast. So this amyloplast is a type of leucoplast. So proplastid can become leucoplast, chromoplast or chloroplast based on the need of the specialized cell. Proplastid becomes into chloroplast for the purpose of photosynthesis process. So the proplastid differentiates into chloroplast. Flattened vesicles develop. The proplastid may or may not have the inner membrane and plastid envelope, and while developing into chloroplast, proplastid develops inner membrane. The inner membrane becomes flattened vesicles which is developed from the inner membrane of the plastid envelope, and they eventually become thylakoid and grana and stroma thylakoids. The thylakoid system of this mature chloroplast appear discontinuous with the envelope and resulting into the mature chloroplast to perform photosynthesis process. The proplastid becomes chloroplast especially in fruits, flowers which are of different color rather than green color. So chromoplast is responsible for pigment synthesis and storage of pigments. So proplastid becomes chromoplast by having the granules of pigments, by having the carotenoids and different types of pigments embedded inside it. Proplastid can also become leucoplast, which is colorless, and these leucoplasts are of three types: amyloplast, eleoplast and proteinoplast. Here amyloplast is there. Amyloplast is the storage organelle of starch in the plants. So proplastid can become leucoplast. There is another deviation or from this image. There proplastid becomes etioplast, where the proplastid develops into etioplast in the case where the plants are grown in dark. They have yellow chlorophyll precursor pigment, protochlorophyll, instead of chlorophyll. When the plant is exposed to light, this proplastid which has been become into etioplast, that will rapidly change into chloroplast. It come like this only, and it becomes chloroplast by converting this precursor chloroplast. So chloroplast is most important member of plastid family, occurs in all photosynthetic eukaryotes. It is a site of photosynthesis. It is a double membrane which encloses a fluid filled region called stroma. Fluid filled region is known as stroma. In stroma, there is a complex network of stacked sacs. Each stack is called grana, and each of the flattened sacs which make up the granum is called thylakoid. The thylakoid membrane encloses a fluid-filled thylakoid interior space, contains photosynthetic pigments. Grana, which is interconnected by unstacked stromal lamella. There are lipids found in thylakoid membrane. About 80% of lipids are uncharged mono and digalactosyl diacylglycerol, and only 10% of phospholipid. So this image suggests the origin of chloroplast according to the endosymbiotic theory, where the early eukaryotic cell, which was unable to prepare its own food, engulfed this photosynthetic bacterium, and this photosynthetic bacterium evolved inside the early eukaryotic cell, making the eukaryotic cell capable of photosynthesis in a process called symbiosis. The first type of plastid is etioplast, which has been developed by proplastid in dark grown plants. They have yellow chlorophyll precursor pigment, protochlorophyll, instead of chlorophyll. When exposed to light, etioplasts rapidly change into chloroplast by converting this precursor to chlorophyll. Then is chromoplast, which is responsible for pigment synthesis and storage. They are rich in carotenoids and responsible for the yellow, orange or red color of many fruits and flowers, as well as of autumn leaves. Leucoplast, which is a colorless non-pigmented plastid, acts as a storage organelle. Based on the kind of substance they store, they are classified into three types: amyloplast, which stores starch; eleoplast, which stores fat; proteinoplast or alluroplast stores and modifies the protein. In our next class, we will be learning about transport of proteins into the chloroplast. Thank you.